webinar is Pancreatlas, mapping the human pancreas in health and disease. Today's webinar has an exciting group of presenters, and they will be sharing with you the work that they've been doing for the last several years. I know that some of you have joined us before, but to those of you that haven't, I'd like to give you a little introduction to HERN and what HERN is about. To the left, you basically see HERN. It is a large network made up of several different groups. And this was initially funded by, and still is, funded by NIH in 2014. And there were two major objectives: trying to understand why beta cells are lost in type 1 diabetes and figuring out strategies to prevent it and or to treat it. Now, the network that you see here is not the ship hasn't sailed. The network isn't closed, and we're evolving all the time, meaning that there are opportunities that come up, RFAs, funding opportunities within HERN. And in fact, there are two opportunities today that are on the streets, if I can say that. The first is called the Gateway Award Opportunity. It's an R03 grant mechanism by NIH, and that allows people to come in not only to HERN, but to other networks. So you as the investigator choose which network you want to come into, but that's one opportunity to join HERN that's available to you now. Another opportunity is through this initiative called PancBase, which is a program trying to draw together data sets and databases, really from all the different initiatives that have been happening in the field. So if you're interested in those, or if you're interested in other initiatives that come up, you can find that on our website. Of course, there is a highlighted here in blue, there is a funding tab. You can check out what we've got there. But today we're going to highlight for you one of the resources that that is in HERN and work that has been happening in HERN through the collective efforts of the group that will be presenting today. But there are a lot of other resources available, and we've highlighted these resources, including today's talk. So you don't have to remember everything. It'll be recorded. You can come back to visit it whenever you want. But these are all cataloged on our HERN YouTube page. So check that out as a resource and other things on the website. So the topics that we are going to learn more about today are here, and you can read those. I will remind you, I think most of you should be muted during the presentation. You might want to turn off your cameras as well. You can put questions in the chat, so I'll moderate the questions toward the end. But you can also ask questions yourself, and I would encourage you to do that. So what I'm going to do now is I'm going to stop sharing my screen, and I'm going to ask Diane Saunders to share her screen. But while she does, I'm going to introduce our speakers to you briefly. So one of our speakers is Dr. Marcela Brissova, and she is a research professor of medicine in the Division of Diabetes, Endocrinology, and Metabolism at Vanderbilt University. She's the director of the islet procurement and analysis core there. And she also directs the Human Islet Phenotyping Program. And that might sound familiar to some of you, because I suspect that a number of you get human islets from the Integrated Islet Distribution Program. Well, her efforts and those of others are doing some functional phenotyping work for that program. Dr. Brissova is funded by NIH, JDRF, the Helmsley Charitable Trust, and in fact, she's also involved in several projects within HERN, just as Dr. Saunders. And Dr. Saunders is a research assistant professor. She's also in the same division there at Vanderbilt. She has a PhD in molecular physiology and biophysics, and double majored in biology and art and visual culture, which would explain her her creative flair in the art. She's the one that designed that figure that I showed you just a couple slides back. Dr. Saunders is also the co-scientific director of Pancreatlas, which we'll hear more about in a minute. And then last but not least is Dr. Cartailler, who received his PhD in biochemistry, biophysics, and molecular biology. And he is the director of creative data solutions and also directs informatics at Vanderbilt University. He's a computational biologist and data scientist with 20 years of experience in software and database developments. He's even started a few different companies to help convey the complex data, how to share complex data in simpler ways. So with that, I'd like to turn it over first to Dr. Saunders.
Wonderful. Thank you, John, for that kind introduction. And on behalf of the team, I want to thank Hearn for inviting us to present today and everybody who's logged on to learn more about us. So just briefly, our presentation today is going to be divided into 4 main sections. So first, I'll give you an introduction to the kind of rationale behind creating Pancreatlas. Then we'll dive more into the technical side of things, give you a demo of the data that's available, including some of the relevant connections to other Pancreas-related programs. And then we'll wrap up with future plans and hopefully get some input from those of you in attendance. So feel free to submit questions in the chat during the presentation, and we'll hope to have some good discussion at the end. And one of the things that we think really makes Pancreatlas a unique resource is that we created and developed this resource within a multidisciplinary team from the very beginning. And that includes obviously myself, JP, and Marcela. So as John mentioned, I have a biology background and an interest in design, and JP is a bioinformatician and database developer. And Marcela, as many of you know, is a leader in the human islet and diabetes research. And so we really think that having developers and biologists in the same room from the very beginning has been crucial to our platform's success. And I think you'll get a sense of that as you hear from both JP and Marcela later on. So many of you are probably already quite familiar with the pancreas, but for those who aren't, the pancreas is a bifunctional organ that is part of both the digestive and endocrine systems. It's situated in the body between the liver, stomach, and intestines, and the vast majority of the mass is composed of acinar cells, which produce digestive enzymes secreted into a branched ductal network emptying into the duodenum. And then scattered throughout the pancreas are small clusters of cells called islets, composed of endocrine cells that secrete hormones to regulate blood glucose. Given its bifunctional nature, diseases of the pancreas can be quite varied, and they include obviously diabetes, chronic pancreatitis, pancreatic cancer, and cystic fibrosis. So many organizations, funding organizations, including the IIDP, support research programs to identify causes and treatments for these conditions. In the past decade or so, there's been a ton of rapid technology development and increased availability of human tissue for research. And so both of those factors have led to establishment of numerous programs to characterize the human pancreas at an unprecedented depth. So I'm showing some of these on the slide here. And so nPOD, for example, is centered around disease states such as diabetes, while others focus on pancreas development or creating a reference organ or a reference of the organ across the lifespan. And since the pancreas isn't easily biopsied, human pancreas research really relies heavily on accessing tissue from deceased organ donors. And nPOD has done a tremendous amount of work to establish partnerships with organ procurement organizations, a network of them across the United States. And this infrastructure, it really ensures that researchers have access to high quality organs from individuals of diverse backgrounds. There's just some quick statistics about some of that work that's being done throughout the country. So many different data types are then generated from these pancreatic tissues, including functional characterization and various omics profiling. But our phenotyping efforts at Vanderbilt, along with several other programs I mentioned, have focused on acquiring cross-sectional imaging data to better understand tissue architecture and spatial relationship between cells. So what this means in practice is analyzing hundreds of tissue blocks in various formats, generating thousands of images in different modalities. And these include the three-dimensional different modalities, traditional immunofluorescence, histology, as well as multiplexed immunohistochemistry. And so a few years ago, we began thinking about how to compile all of this data in a meaningful way. that would essentially link the de-identified clinical information with high-resolution images that could be easily accessible by those in the community. And so we started with data from the 4 programs that are shown here, and we conceived of an atlas, which eventually became named Pancreatlas, that would serve as kind of an imaging resource detailing the human pancreas in both health and disease. Our first task was to address some of the challenges I mentioned previously, such as image quantity and size. We wanted to make sure that despite those variables, we had a user-friendly interface. We also knew that we needed to be able to support a wide variety of image modalities and file types being generated. And we knew we wanted to capture pertinent metadata so that people could analyze and interpret these images in the correct context. And then finally, a lot of times you see images as, little teeny panels within a figure of a publication. And so we really prioritize the ability for people to interact and really interrogate those images at a more high resolution. So to explain how we tackled these goals, I am now going to hand it off to
Okay, thank you, Diane. So hello, everybody. Thank you for joining us. I'm JP Cartailler, and as Diane indicated, I'm going to give a brief overview of some of the work we've done in the past year or so for adding global search functionality to Pancreatlas. And part of that process has also been focused on harmonizing our metadata so that can become a reality. Okay, here we go. So, for Pancreatlas in the very, very beginning, and again, as Diane pointed out, sort of having a very mixed team of individuals from very different backgrounds, we really wanted to strike a balance between having a tool that was usable for biologists and non-technical users, but a tool that also over time would provide the required complexity and flexibility that developers might want to use to access the data or the metadata or both. And again, some different timelines on this. We knew from the perspective of requiring our funding obligations and getting the wow factor out there, that we're really focused first on building the user interface and building a nice, easy to use website. And we've been working ever since, and of course, continuing that process, but also now starting to add more tools and mechanisms for developers or machines to use the data. And in striking that balance, we really had a set of core principles we started with. One, we really did not want to reinvent the wheel, so we just went out there, really just get a good gauge of what all the tools, or both commercial as well as open source, free versus non-free, were available. And we really thought about what is it that we want to actually build versus just use out of the box, and how can we make those things work together. Again, we really wanted to have user-friendly and online-only tools. As indicated earlier, with all these different imaging modalities, the hundreds of thousands of images, nobody really wants to sit down and download all these images only to find out that they don't have the proper viewer because it requires some proprietary viewer or plugin or some combination thereof. So again, from a usability perspective, we really want end users not to have to download or install anything, just to use their web browser, which everybody has. And of course, we wanted to support any image format. That's a very large claim, but we definitely found that to be possible through the Glanco Bioformats library. And again, not reinventing the wheel. And also, we really felt that in order to properly control the data and the metadata, we really needed to go through a curated and supervised process. And I'll speak a little bit about that as well. So again, we started off with simply imaging, and the platform, after doing a survey of what was available, we decided to license a software called Omera Plus, which is developed by Glanco Software. Omera Plus is actually a commercial version of the open source OME product that anybody can get. So you can go download Omera and use it out of the box, but Omera Plus comes with additional libraries, including the Bioformats library, as well as a proprietary image viewer. And for us, this was important because we really did not want to reinvent those tools. It would have been much too costly, and it would have taken years and years and years if we could even, and again, there's no point reinventing the wheel. So we installed Amera Plus within the IT infrastructure at Vanderbilt Medical Center. We have pushed data from the LIMS into this environment. And of course, we've connected various sources of image, local image storage that could be read from Amera Plus to actually ingest the data. And over time, we built this custom web interface, which we hope that many of you have already accessed at pancreatlas.org, or will do after this webinar. And again, Amera Plus comes with a proprietary viewer called Path Viewer. It's really a fantastic viewer, and one of the key things about it is it allows you to view more than 4 channels at a time. So you can really look at something with 30, 40, 50 channels. Granted, you can't look at 50 channels at the exact same time, but the cutoff we have it, we've configured it to be about 30 channels at a time. Any more, it starts becoming a little bit too data intensive in the back end, but typically that's usually sufficiently enough for most people to work with. And as a result of putting, these ready-made available tools and building some novel tools on top of it and connecting all these dots, for the last few years, users have been able to access this interactive, full-featured image viewer that they can interact with, zoom in, turn channels on and off, turn panels of channels on or off, and really just look at all this data through very carefully curated points of entry to a variety of large data sets that are either contextual from phenotyping efforts, or also just additional data that were associated with the particular papers that were never published. And that's actually something we're always on the lookout for, to incorporate other people's data that may not make it to journals, because there really are very few mechanisms for making this available. So again, coming back, we wanted to leverage Glencoe's software capabilities through Amera Plus, and it really is no joke that the bioformats library allows us to read and access data from over 160 different file formats, and it just works out of the box. I mean, this is something that's definitely worth it, and has made our lives much easier. And then the Amera system itself, it has a backend database that has a very large software stack, and essentially allows us to bring data in to manage within Amera and not have to deal with that whole image management issue. It just works out of the box. So what we did is we now have to get data into it, and we wanted data to be properly annotated. So within this team, we developed a spreadsheet-based metadata import system. It doesn't sound all that novel, but it works really well. So of course, we constructed a careful spreadsheet with the types of annotations that we'd be interested in capturing alongside of each of these images. And we have these color-coded here, and these are sort of all the researcher-defined variables. Obviously, we have a set of common variables we always want to populate as much as possible, and then there could be additional variables that can also be loaded as into the key value store. And again, in the spreadsheet, every row of metadata corresponds to a single image source. And we have an import script that wants to just provide this very simple spreadsheet, which of course references a location to where the image data actually resides. All that data gets imported by Amera. It can spend a few hours to a few days just churning through it, processing, building image pyramids, so on and so forth, and eventually storing all that. And then Pancreatlas, which I've shown here on the right, can actually then go in to the Amera API and pull out the data that it needs in order to present the data within this custom web interface. And as you can tell, again, from the color codings, a lot of these researcher-defined variables you have in the spreadsheet are actually shown directly on the user interface as a set of filtering options. And of course, you can filter things like, for example, disease status, disease duration, age, sex, modalities. There's lots of things that you can filter through. And over since inception, we've actually collected and curated over 2,000 images on pancreatlas.org. And these are available through a set of collections that we've shown here. Again, some of these are from large-scale phenotyping programs. and several are also from manuscripts as additional data supporting those manuscripts. And very recently, we also had a new collection that was imported from the Exeter Archival Diabetes Biobank. This contains really a very large set of data, very, very high resolution. I believe they're 40x microscopy, so you can go in very, very deep dives. And it was definitely some new challenges for loading that data, but we strongly urge you to go check that out. So one new feature we've been working on, and this is still work in progress. So we've collected all this family of collections, which each have their images, and we now have a mechanism for actually searching and filtering through all these collections. So if you go to the pancreatlas.org homepage, we have it. Hopefully you can see it should be right front and center. You can just go in and essentially search by marker. So for example, you can start either there's a drop down to get an established list, or you can start typing in your favorite marker. Hopefully there's a match. And once there is, you just click on search, and that will build the image grid that in essence retrieves all the images in Pancreatlas that are, that include the insulin marker as, one of the channels. And you can see here on the left side, these are all the different data sets. So essentially these are the, all these otherwise known as collections. And on the right side, you can see all these image panels. Each panel represents a single image with sufficient metadata, we hope, to allow you to kind of further explore. And of course, if you click on those, and we have a walkthrough later, you'll be able to get a larger image, in the context of Path Viewer. But within this context, we're now, just out of insuling, we're filtering out or viewing 142 images out of about 2,200 actually. And then of course you can go in and further filter. In this case, we've highlighted, we wanted to, highlight type 1D. So we've further selected that. So this is 48 images that are, that have an insulin channel and are type 1D origin. So there's lots of ways to filter through this data. So we encourage you to check it out and give us some feedback. And with that, I'm going to pass it back to Diane. Thank you.
To give you a quick tour of the website, pancreatlas.org. From the homepage, you can either click image collections or the down arrow to view all data sets currently available. If you'd like more context, select collection details to read more about the project, or you can simply choose a collection and begin browsing. Once in a collection, you'll see a grid of available images, each of which has its own card that lists important donor and sample characteristics, as well as the different markers visualized. On the left here, you see metadata filters, which will refactor automatically to give you available images as you select different attributes. Clicking on the magnifying glass launches a modal preview, which enlarges the image thumbnail, gives some additional sample information, and then allows you to navigate directly into Path Viewer, which is Glencoe's web client for exploring images. So this particular image was acquired using Codex technology, which is a fluorescent immunohistochemistry-based multiplex system. This is a cross-section from a non-diabetic adult donor, which is full resolution at 20x magnification and contains 28 registered channels. So notice that the image is loading almost instantly despite being more than 25 gigabytes, meaning that you can immediately browse and interact with multiple images within a given data set without the friction of long download times. And instead, Path Viewer really lets you dive straight into the data. So first, I want to just briefly highlight the list of markers to the left. These are grouped into biologically relevant categories. So not only can I pan around the image and explore areas of interest, I can also toggle on and off channels depending on the cell type that I'm interested in. So for example, right now the endocrine cells, hormone producing cells are shown, but if I wanted to, I could add in a vascular marker to visualize islet capillaries, or I could choose an immune marker in order to understand the proximity of macrophages to various structures within the pancreas. So you can see there's ducts highlighted here in cytokeratin in orange, and then you can also see the ecadherin that is marking individual acini. The flexibility of Path Viewer also means that in the future, we'll be able to import dynamic object level masks like cell type or cell neighborhood annotation, which can provide even more context to the raw images. A couple of other things I just want to briefly mention. From the header menu, you can easily access our nomenclature tab, where we've provided accessible descriptions of terminology and have tried to really standardize metadata across our collections. And this can be particularly useful if you're looking for markers to label a specific cell population in the pancreas. And we've even linked to the Human Protein Atlas in pertinent cases so that you can get even more comprehensive information. In addition, we hope that our resources page kind of acts as a one-stop shop for other resources. This is particularly designed for trainees or investigators who may be new to pancreas biology and curious about what is out there. To give you a quick tour of the website, pancreatlas.org. Sorry about that. Okay. So what I want to talk about now is a little bit more HERN focused and pancreas program focused context. So you're probably wondering why host data that's already available elsewhere. We talked about a couple of other phenotyping programs, including some that are supported by HERN. And really, I think the value of viewing images in Pancreatlas, aside from obviously the great capabilities of Path Viewer, is that you can view this data in the context of a lot of the different efforts, which otherwise you would have to kind of go back and forth hopping between different databases. And so two that I'm going to briefly share with you today are PancDB, which is a database that is holding data that is generated by the Human Pancreas Analysis Program, which is part of HERN. And then the other one I'm going to mention is the Integrated Islet Distribution Program and the Human Islet Phenotyping Program, which is part of characterizing which characterizes all of the human islet preparations that are distributed for research across the country. So to start with PancDB, some of you may be familiar with this interface. You can also reach it from the HERN website as you can Pancreatlas. But essentially, if you are browsing through the donor information, the donors that make up this database so far, you'll notice that there are individual data set types that are listed across when you kind of maximize the window for any given human donor. And so when you see either imaging mass cytometry or histologic imaging in one of these windows, you'll actually notice that in addition to getting a preview of the image and some pre-curated combinations of markers, you'll also see this button that will take you over to our website so that you can interact and see more clearly the different channels or different combinations of markers in those images. And so here I'm just illustrating, it's going to take you, that link will take you directly to the Human Pancreas Analysis Program collection. And so you can see from this screenshot here that you're going to be able to browse both histologic images, H&E, but also that you'll get a chance to really interact with the imaging mass cytometry images, kind of similar to what the Codex one that was shown in that site tour. And then also we link back out to the HPAP database. So again, if you're browsing images in Pancreatlas and you want to find out more information or maybe other data sets that are available from that donor, you can click on the hyperlink that is part of the program ID, and that'll take you right back to PancDB and to be able to look at some additional data sets. And then in a similar fashion, we worked with the HIP database team at IIDP. If you are a registered investigator with the islet distribution program, when you log in and you see all of the different islet preparations that you've received, one of the options at the end of your screen will be this button with our icon on it. And so clicking on that icon will take you back to Pancreatlas. These are actually islet embedded. They're islets embedded in a collagen gel, so they kind of provide this really cool histologic image of islets, but that are suspended in a way that you can really appreciate the composition of the and arrangement of the cells. And so again, this will take you right into the IIDP collection within Pancreatlas. And if you want to go and find out more, you can click on the IIDP link. And if you are an investigator that has access to that data, you'll get to kind of see what other phenotyping information is available from that data set. And then the last thing I'll just mention is that we're really excited about a lot of other potential cross-links between different programs. And so one that I've been interested in through my work with the HubMap Consortium is this effort to really create a common coordinate framework of the human body, including a lot of the human organs that are being analyzed for various phenotyping efforts. And so you'll notice that already within Pancreatlas, we're trying to give very critical information about where the sampling occurred within the organ to give you some context when you're interpreting the image. And so what we are envisioning in the future is that actually right now you just get the information in the form of a label, but actually HubMap has built this really incredible interface where they're allowing people from all different programs, phenotyping programs, to actually register the location of samples that have been analyzed. So you can see here in this screenshot, there's some from the GTEx project and some from HubMap itself, but you can go to this website and it's really cool. You can actually like look around at the human pancreas in 3 dimensions and kind of see where the data was sampled from in each of these particular cases. So we're really excited about other potential opportunities to take advantage of other data sets and other programs that are out there. Okay, and so now what I'm going to do is I'm going to just briefly show you how we are thinking about using this resource essentially to better understand the data that they're collecting or that their colleagues are collecting in the context of other research efforts. So here I'm just displaying the Human Pancreas Analysis Program collection. And so, for example, if I was particularly interested in type 1 diabetes, I could start to drill down at a particular subset of these samples. So let's say, for example, I wanted to know, or I wanted to look at images that were from donors with a pretty short duration of type 1 diabetes. So you can see here, I'm going to be able to explore 24 images in this collection from HPAP, and there's a lot of cool data here, but it does look like I'm limited to 3 or 4 different donors. So, for example, if instead I wanted to query across the entire Pancreatlas collection, I could go into the Explore All data sets rather than starting specifically from the HPAP data set. And now, if I apply the same filters, so I want a type 1 diabetes, and I want something that's relatively short duration of type 1. Now all of a sudden I have 233 images rather than just a handful. And so actually you can view these and interact with them in Path Viewer like I demonstrated, but now all of a sudden we're able to look at the Exeter Archival Diabetes Biobank collection in the same context as HPAP. And so I just wanted to illustrate that example as kind of an option for if you're already collecting data and you want to see it in kind of a more specific context from a lot of these really incredible phenotyping programs together. Okay, so now I'm going to head back to our presentation. And okay, so that was just one example, but I also wanted to give you a couple of more examples of things that you might be thinking about or that your colleagues might be interested in. So one example would be, how does the architecture of the human pancreas change during the first decade of life? We have a whole collection based around human pancreas development. Another thing people are very interested in is what changes are occurring with type 1 and type 2 diabetes. Also, particularly some programs are curious about what is the real range of normal or quote unquote normal pancreata. So that's something that, again, combining images from different sources really helps to get at that question. And then finally, with the development of more multiplex methods, you can now really be able to search and filter for some markers that are specific to your particular cell type of interest. Okay, and with that, I'm going to turn it back over to Marcela.
All right, let's see if we can do this. So in the last segment of this presentation, I would like to highlight our future plans, and we would love to get feedback from the community on Pancreatlas. So I will conduct a brief survey at the end. Okay, let's see if we can navigate this. All right, So as we are looking ahead, I would like to highlight how we are thinking about technical advances and then feature-based development. So on the side of technical advances, what we would like to do is build a knowledge graph to put knowledge around the pancreas and mapping effort under one umbrella. We would like to improve compliance with FAIR principles, something that NIH is going to implement starting next year. And also we are working on harmonization of metadata fields with programs such HPAP. HPAP was really instrumental in developing really thorough annotation along anatomical structures, cell types, and biomarkers. Now, on feature-based development, I would like to highlight two areas, and it's really a connection of imaging data with other omics data sets, and then cloud migration of Pancreatlas that would enable also some analysis opportunities. So, as we are thinking about pancreas mapping efforts, really the power here is in multidisciplinary teams and ability to integrate these very rich data sets. So starting with imaging data, where you can appreciate spatial relationships of cells and overall tissue architecture, then overlaying that with physiology and transcriptomic and chromatin signatures, you can then connect that to genetic risks and variants, and which in the area of diabetes, these multidisciplinary integrative approaches are now really paving the way to cellular attribution of GWAS loci, for example, and how do they relate to developmental timeline, disease state, and also different ancestries, for us to gain more understanding into the mechanisms underlying signatures of personalized signatures, really of genetic predisposition. And so with that in mind, Pancreatlas platform was really built as data agnostic platform. So you saw a large amount of work that's been done in terms of incorporating imaging data sets to Pancreatlas, but looking forward, we would like to bring omics resources. So currently using specialized viewer, path viewer, you are able to view imaging data sets, and again, using a specialized viewer for single cell data, we would like to bring in single cell data sets, and just like images, they have their individual cards. Also, single cell data sets would have their individual cards in the Pancreatlas. We are also currently working on uplift of Pancreatlas into the cloud, which will increase scalability of our platform, and also it will make it easier for external collaborators to upload their data sets, and will ease the access to raw imaging data and enable analysis of imaging data in the cloud. So we are making progress there, and also in terms of connecting programs that are working on different ends of these pancreas mapping efforts. Diane very nicely walked you through 4 programs that are currently connected to Pancreatlas, and I would like to highlight this 5th one, which is Exeter Archival Diabetes Biobank. This was international collaboration with the group in the UK, and we were able to connect a really unique collection generated by Dr. Follis that is focusing on childhood early onset of type 1 diabetes, really unique data that paved the way to be able to start diving into processes and pathologies of the pancreas early on. We are expanding these international collaborations through Human Cell Atlas initiative to connect with group ESPAS based in Europe, and we're hoping to connect also to programs like HPAP that Diane mentioned earlier and others. And so finally, acknowledgement slide. This is our last slide of the presentation. I would like to recognize our Pancreatlas team, which is really wonderful group of individuals to work with. I'm very honored and privileged to be part of this team. I would like to acknowledge our funding agencies, especially Hensley Charitable Trust, our collaborators, in nPOD, HANDEL program, HubMap program. Most of them are based at the University of Florida. And I would also like to recognize contributors here in the US and in UK. And finally, I would like to express my gratitude to organ donors and their families, without whom this work would not be possible. And before we go to question and answer section, I think it would be great if we could get your feedback, and you can navigate to this menti.com site. Here is the code for it, or you can just quickly scan the QR code to be able to take the survey. It's a very brief survey, 2-question survey, so it shouldn't take too long. And before I go, one last thing. Let's see if I can go there. Yes. We would love to connect. So if you have feedback or suggestions, please reach out. If you would like to share your imaging data sets, please connect with Diane. And we are currently have positions available for that database work. So please reach out if you would like to work with us. And with that, I'm going to turn it over to John Cadiz.
Thank you so much, Marcela, and thank the three of you for putting this together. I must say that was very smooth. There were no technical difficulties at all. That was wonderful. If you do want to pose questions to any of the speakers, go ahead and take yourself off mute. And if you can, raise your hand. It'll just help me stage who was first in line. So I'm going to check the chat, and by the way, Diane has included that link that Marcela highlighted for you, so you can just click on that, go to the chat, and take that quiz, or the quiz, take the thinking much differently. Okay, take the survey. It's 2 questions. You'll get an A at the end, I'm sure. So if you can do that, I'm sure that they would be helpful. I don't see any there, but we'll give people in the audience a chance to queue up their questions. I do have a couple questions. Actually, Diane, for you first, if I've gotten this right, very impressive, over 2,000 images. I've been looking, as you've been talking, I've been looking through some of the filtering criteria on the left. So is that right? 80 markers and 351 donors represented in all of these images? Quite impressive. So for one particular case, Diane, you said 40 channels. Is that in this collection? I know you can do a lot. In this collection is 40 channels. You have 40 channels worth of data?
Yes, I believe a couple of so if you go to the HPAP collection or you filter by IMC modality, I believe some of those images have, close to 35 or 40
Wow.
channels.
That's very nice.
Yeah, and you know, as JP mentioned, you can't actually turn them on all at once because your eye can't even like understand that many colors.
Right.
But you can do like 8 or 10 at a time, and choose. Most importantly, I think you can choose which ones you want to see, so you don't have to rely on whoever made the figure panel or whatever. You can pick what's interesting to you and change them out in real time.
Yeah, I hadn't seen that, and that's a really nice new feature where you can essentially sort by marker. So kudos to you for that. A second question I have, I can keep going. Let me just make sure that I don't see anybody with their hands raised, and I don't. I think the second question might be to you, JP. During the presentation, the metadata was described a lot, and you showed how you were able to upload the metadata, and a lot of us in the field work in different programs. So we have our hands in, HERN or nPOD or different things. Is there any way that you plan in the future by API or some other method to allow extraction of the metadata? It's not practical nor feasible to do it for the images, of course, but for the metadata, it might. And have you all been thinking about that in any way?
Great question. Thank you. In short, yes, we've been thinking a lot about this. So we have an API, it's just not open to the public, and we need to, what we really need to do now is, because we have, I think enough images, enough metadata, and we've gone through several rounds of re-standardizing the metadata, and we're not quite done with it yet, because we are now injecting a lot of essentially, ontology terms and IDs
Yep.
in the way we have a common vocabulary that everybody else can use, so in addition to maybe some of the internal vocabulary that we use. But we do need to finish that, and we need to essentially write up documentation on how to use the API, and then we'll definitely get it out. And then to add to that, Marcela mentioned that we're currently uplifting, quote unquote, we're moving our infrastructure from institutional VMs to putting in our case, in AWS. All the data is going to follow. So we want to set it up so that way, if people want to access the raw data, they'll be able to do that directly through an S3 bucket to S3 bucket transfer. And if they want to move it to a different zone, they'll have to, essentially pay for it, but that won't be our problem. It'll be, here's the data, it's yours now. So I think that in combination with the metadata will definitely allow others to start doing more computation in this data, which is something I think in the next 5 to 10 years will be very exciting.
That's terrific. That is really, really great. I'm happy to hear all those plans. Okay, any other questions from the audience at all? I can tell you that this group here is quite helpful in getting you to connect with the data and using the data. In fact, you're also involved in other initiatives whereby there are challenges going on for people to use this data to get new insights. And I know that you're involved in some of these other initiatives as well. So there are video resources and other things if people want to learn more, but I think this is a great resource, and I don't see any other questions. So I really would like to thank the three of you. I know it takes a lot of effort to put this presentation, and it's not easy to have 3 speakers and to do it so nicely. So thank you very much for your time and effort and for what you're doing in the field.
Thanks, John. And I'm just going to make another plug. The survey is in the chat, the link. And sorry if you already answered, thank you. I fixed where it should advance to the second question now. It's just an open-ended feedback. But if you haven't already, it's totally anonymous. So please, if you have 30 seconds, click on that link in the chat and it'll take you to the survey. We want to be able to build features in the future that the community is interested in. So this really helps us get an idea of what you all are wanting. So feel free to reach out to us too if you need any help. I'll put the email address also in the chat.
Okay, terrific. So we will we'll stay in here just for a minute or two so people can grab that email address and link. But otherwise, again, I'd like to close off this webinar and thank everyone for presenting.